Good morning, welcome to this class on Neuroscience of Human Movement. This is part 2 of our discussion on membrane physiology. So, in this class we will be talking about ion channels, different types of ion channels. We will give a couple of examples of these ion channels, voltage gated ion channel and ligand gated or chemically gated ion channel, and we will be talking about diffusion potential, and we will be at least defining the equilibrium potential. So, we discussed the case of plasma membrane in the previous class. Suppose a substance or an ion has to be transported from say one side of the cell, say from the outside of the cell, let us say this is a cation, this has to be transported from outside of the cell to inside of the cell. Suppose this has to happen, there must be a channel, there must be a protein that must allow this transport to happen. Note that these channels may be selective, and so in general these channels are selective, and this selectivity may be due to size or may be due to type of charge. Some channels may allow only cations, some channels only may allow sodium but not potassium, some channels may allow cations but not anions, etc. So, also note that these channels are distributed along the length of the plasma membrane. So, that means that there is spatial distribution of these channels. Also, if this channel is open, the probability that this ion is going to get transported say in this direction, or suppose there is a different channel here and a different ion gets transported in that direction, say for example. The probability that this channel is open governs to a large extent whether that ion will get transported or not. So, this probability is usually represented in terms of permeabilities, or to use a phrase, to use a term from physics and electrical engineering, conductances. So, if a channel allows, if the probability that the channel is open is very high, then the channel has high permeability for an ion and or high conductance for transport of that ion. Also note that this probability is a function of time. It may be open at certain times but not be open at other times. So, that means there is also a temporal distribution of the opening and closing of these channels. So, there is both a spatial distribution and a temporal distribution, and that affects permeabilities. And note, not all channels allow all types of ions to pass through. So, there is also selectivity in terms of which ion passes through which. So, what you have is a relatively complicated situation where different channels allow different types of ions and they are open at different times depending on different conditions. So, how do we study this? And studying this could somehow inform us about the function of the cell membrane itself, right. So, as I said, ion channels may be selective, they may be selective based on charge or size, they may open at different times, they are distributed along the cell membrane. This is the spatial distribution part, this is the temporal distribution part, and this is the selectivity part, right. So, an example of channels is the voltage gated sodium channel. Here is the case of the voltage gated sodium channel. So, this is opened or closed, this is controlled by the potential difference across the membrane. When the potential difference reaches a particular point, also called as a threshold, when a threshold is reached, this channel undergoes a conformational change, or this channel undergoes a change in the structure in such a way that it allows or disallows the ions to be transported. So, let us take the case of sodium getting transported in this voltage gated sodium channel. Okay, this is a voltage gated sodium channel. So, in this case, as soon as a threshold potential is reached, what happens is that a command to two gates. So, this channel has two gates, one is called as an activation gate and another is called as an inactivation gate. Both of these gates are given command to change from their current position. Please note what the current position is. The current position is at rest, the activation gate is closed and the inactivation gate is open. This is the rest state. And when threshold is reached, both of them are given command to change from their current state. That means what? That means the activation gate must open and the inactivation gate must close, but these two are timed differently. These two processes are the closing of the inactivation gate and opening of the activation gate are timed processes, they happen at different timelines. It turns out that opening of the activation gate is a rapid process and closing of the inactivation gate is a relatively slow process, right. Both of them are triggered simultaneously, but one is a fast process, the other is a slow process. So, this will effectively result in a small amount of time during which the channel is open, and during this time a lot of sodium can enter inside the cell from the extracellular fluid, right. So, the on top is the extracellular fluid, in the bottom is the intracellular fluid. During the brief time during which the inactivation gate is closing and the activation gate has already opened, during that brief time gap lot of sodium enters inside, and this is a crucial event we will discuss in future classes, right. The other example is the transmembrane protein or the channel called the ligand gated channel. So, this involves a particular chemical binding to a particular binding site like here. So, once this chemical binds to its binding site, this channel opens and allows transport of sodium, for example, right. The classic example is the case of the nicotinic cholinergic receptors in the neuromuscular junction, right, which we will discuss in great detail in future class. Okay. So, this means when these ions are transported from one side, say this is sodium and this is getting transported from the extracellular fluid to the intracellular fluid, say for example, when this happens, it creates a charge separation. What effect does this have is of importance for us in discussion. So, let us see what would happen. Here we have taken the case of a potassium channel. Suppose there is a potassium channel that is open, and when it is open, note potassium is present in great quantity inside the cell when compared with outside the cell, right. So, as potassium travels in that direction, so when the channel is open, potassium is going to slowly go out from inside of the cell. As it is traveling from inside of the cell to outside the cell, it takes one positive charge from inside to outside also. So, basically the concentration gradient is in that direction, but as the amount of potassium builds up on the outside, the potential difference prevents the buildup of more potassium outside the cell. Basically the electrical potential gradient is in that direction. That is why these two are in different colors, right. This is the electrical gradient and that is the concentration gradient. So, in two different colors, right. So, these two are against each other. That means there must be a point at which some sort of equilibrium must be achieved. What is that point? That is of interest for us. We will discuss that in future classes. Okay. So, if a cation diffuses down a concentration gradient, a potential difference is developed, as I said in the previous slides, and this potential difference start resisting further diffusion of cations in the same direction. So, that means as the amount of cations build in one side of the cell, that means that more and more cations cannot go, or the ease with which the cation can move in the same direction is reduced, right. At some point the potential difference does not allow any more cation to enter in the same direction. So, basically what happens is the diffusion completely stops or the transport completely stops, although there may be concentration gradient. The concentration gradient may be present, but the electrical gradient will not allow any more cations to enter, right. So, what is achieved at this state is an electrochemical equilibrium, and the potential at which it is achieved is called as equilibrium potential of that cation, or also called as the Nernst potential of that cation, right. So, we will discuss that in the next class. Okay. Let us consider the case of the sodium and discuss how diffusion potential is generated in the case of sodium. Let us consider the situation where there is a hypothetical membrane that separates two chambers of sodium chloride. In one chamber you have a relatively high concentration of sodium chloride, and in the other chamber you have a relatively low concentration, okay. And the membrane is selective only to sodium but not chloride. So, that means what happens is that only sodium will get transported from the region of higher concentration to the lower concentration, but not chloride. What this will do is that it will create more positive charges on the right side of the chamber or in solution 2, that means effectively making the left side a little less positive or a little more negative. So, it creates a charge separation. At some point the amount of positivity in the right side of the chamber does not allow any more sodium to diffuse through, regardless of what the concentration gradient is. The gradient might still be in this direction, or the concentration gradient might still be in this direction, but potential difference will not allow any more transport of sodium. At this point sodium electrochemical equilibrium is achieved. The point at which that happens is the Nernst potential of sodium, right. Likewise for chloride, suppose I had a chloride selective membrane, the opposite will happen. The right side will have a buildup of negative charges, and the left side will have a buildup of positive charges, effectively creating charge separation one more time. Chloride will get transported, but after some time chloride transport will be prevented, effectively stopping any more chloride transport. The point at which this happens is the chloride electrochemical equilibrium, right. So, in summary what we have seen is that ion channels may be selective in charge or size, and they are distributed along the cell membrane. There is a spatial distribution, and they may be open or closed at different times. So, they may be more permeable or have high conductance to specific ions, or maybe less permeable or have low conductance to different set of ions. And the opening and closing of these channels may be due to membrane potential, or voltage gated, or may be chemically gated, or maybe for example it may be ligand gated, and the potential that is caused due to diffusion of ions is called as diffusion potential, and the potential at which electrochemical equilibrium is achieved is called as the equilibrium potential or Nernst potential. We will discuss Nernst potential in the next class. Thank you very much for your attention.